Welcome to the online training for the Integrated Monarch Monitoring Program, or IMMP. The IMMP is a program to collect data on monarch butterflies and their habitats across their entire breeding range. The data will be used by a variety of conservation professionals and researchers to advance our understanding of monarchs and their habitats, set conservation objectives, and monitor progress toward them. We're excited to have you join this effort. Activity 4 allows us to track monarch survival and parasitism. Participants rear larvae collected from their monitoring plot to assess survival outcome and submit samples to track parasites. This activity helps us estimate larval survival and measure the importance of mortality factors at different times and locations. It also allows us to estimate rates of parasitism. Some common parasitoids we will track are wasps and tachinid flies, shown on the left and center here. Parasitoids typically kill their hosts, here the host being the monarch larvae, and the parasitoids have an adult stage that's free from the host. We will also track the parasite Ophryocystis elektroscirrha, a parasitic spore, shown here on the right, commonly called OE. Parasites depend on their host through their entire lives and don't always end up killing the host. Here we'll discuss how to monitor or test for all of them. Daily care is required while rearing larvae. We recommend rearing up to 10 larvae at a time and no more than 30 from your monitoring plot each year. You do not have to collect every caterpillar that you find from your plot, especially if you don't have the capacity to properly care for them at that time. Collect 4th or 5th instar larvae from your monitoring plot. If you are conducting Activity 2, the Egg and Larvae Survey, collect your larvae from just outside of your plot so as not to affect the density and survival estimates for Activity 2. Taking in larvae from different times of the season is helpful, as some pathogens, such as OE, vary throughout the year. You're welcome to rear monarchs from other areas too, just keep those data on a separate form. Data about monarchs from other locations beside your IMMP plot should be entered into the Monarch Larvae Monitoring Project website, www.mlmp.org. Here's a reminder of what 4th and 5th instars look like. At the top right, you can see the 4th instar's tentacles, if folded down, would extend just below the tip of the head. At the bottom, the 5th instar's tentacles extend well beyond the tip of the head and are so long that they're droopy. Use your Activity 4 data sheet to record the date, location, and instar at the time of collection. Rear the larvae indoors. Monarchs can become parasitized even if containers outside, because some parasitoids, such as wasps, are very small and fit through mesh screens. You'll record the survival outcome and a few attributes related to it. You'll also submit relevant samples, such as the fly, if the monarch is parasitized. Always rear 1 monarch per container. Well-washed deli containers or a mason jar with nylon or otherwise ventilated top work well for rearing. Petri dishes or even small condiment containers work well for smaller instars, and you can move them to larger containers as they grow. Keeping 1 monarch per container is the best way to ensure disease is not spread among larvae, and so that you can track specimens accurately if you're raising more than 1 at a time. Feed the larvae fresh milkweed each day, taking out the old ones and emptying their frass, or poop. When collecting leaves, make sure to locate places where you're pretty certain plants have not been sprayed with pesticides. You can collect a handful of milkweed to keep on hand and retain it in your fridge with a damp paper towel for a few days. To keep the leaves inside the container fresh, it helps to keep a damp piece of paper towel in the bottom of the container. The towel should be moist but not dripping wet. Clean the containers after each use. Use a 20% bleach solution or other option outlined in the program guidebook. A common outcome is survival to adulthood. In this case, after about 10 days in the pupal stage, an adult monarch will eclose or emerge. Allow 24 hours for the adult to hang and harden its wings, and do not touch the butterfly during this period. Then, test it for the parasite OE, as described later in this webinar. The monarch may also die. This could be from disease, such as a bacteria or a virus. Lab testing would be necessary to determine what type of disease, and this program does not have a component to test dead larvae. Record the outcome, such as dead from unknown disease, or turned black and died, and then discard the larvae in the trash. Larvae may also be infected by tachinid flies. Tachinid flies look a bit like houseflies. They lay eggs on the surface of a monarch larvae at any stage of development. The eggs then hatch, and the fly larvae burrow into the caterpillar. After developing inside the caterpillar, the fly larvae will emerge from the monarch, typically in the J or early pupal stage, causing it to die. And then they pupate at the bottom of the container and look like small reddish pills. Here you can see the silk threads that they use to exit the caterpillar. Their pupal case is about the size of a tic-tac. Wait and let the adult fly emerge from that pupal case. Then freeze the adult and send it back to the University of Wisconsin address shown later here and also in your guidebook. Parasitoid wasps lay eggs inside monarch pupae, so you won't have these unless you collect a pupa from outside. A wasp-infected pupa may look grayish-brown, and then 2 to 3 weeks later, dozens or hundreds of tiny wasps will emerge. Place the container in the freezer to collect a specimen to submit. If your larvae is parasitized, submit the parasite to the University of Wisconsin Arboretum. You do not need to submit the monarch larvae or the pupa, only the parasite that emerges. Place it in a pill bottle with tissue. Don't use cotton, as the fibers can get stuck to the sample and make it difficult to identify. Label each container with the information shown here. All of this information is listed in your guidebook. On your Activity 4 datasheet, record data for all monarchs in a season. Then you'll only have to enter one data sheet into a Survey 123 or online at the end of the season. Make sure to include your name and site ID. Then number your larvae as you collect them, such as by placing a label on the lid or the rearing container. For example, here we collect a larvae from our plot on April 29, 2018 as a 5th instar. It emerged from its pupa on May 14, 2018, and we identified it as a female. A day later, we tested it for OE and then released it. That's all the information that you need to record for this successful adult. We also collected, on April 29, 2018, a 4th instar. This instar did not survive. It was parasitized by a tachinid fly, so we write that result in the results column. We wait for those parasites to emerge and then count the number of flies that emerge from the monarch. We record the date that they emerged from the monarch. Our 3rd larvae of the season was collected on May 6, 2018. We collected this as a 4th instar. This died a few days later, so we record that in the result column. We don't know why it was not parasitized. We write unknown cause. Mark your containers with these ID numbers so you can keep track of them. Ophryocystis elektroscirrha, or OE, is a protozoan parasite that affects monarch and queen butterflies and can weaken or kill them. OE appears to vary across different times of the year and different regions. It's higher in locations where milkweed persists through the winter. We know this largely because of data collected by volunteers such as yourself. Project Monarch Health has been helping citizen scientists test for OE for years. They offer a convenient sampling kit from their website. Alternatively, you can use clear Scotch tape and index cards to sample. At the end of the season, you'll need to submit a sampling card and a copy of your data sheet to Project Monarch Health. When submitting OE samples, all of the information associated with the collection, like location and stage, are important to include as well. To test for OE, first put on gloves or wash your hands thoroughly. Hold the monarch firmly at the base of the wing, as shown here. Press the Monarch Health sticker or small section about 1 inch of clear Scotch tape to the bottom of the abdomen. Press firmly enough to collect some scales, which look like black dust, on the tape. Removing these scales does not harm the butterfly. Release the pressure to remove the sticker from the abdomen and stick it to the index card. Remove your gloves and sanitize the workstation. Then label the sample with the identification number you assigned to the Monarch on your datasheet for Activity 4. Here's an example of OE spores among larger butterfly scales. Project Monarch Health will look at all your samples under a microscope to see if OE is present. This can even be done from home with a 100 or 200 time magnification microscope. After you've sampled your adult butterfly for OE, release it back to the area you collected it from if convenient, or release it in an area where the butterfly has readily available nectar resources. You will not have results back from the OE testing at the time of the release, but because OE is a naturally occurring parasite, you should release the butterfly whether or not it's infected. If you find out at any time that your OE infection rate is very high, it may be that you're inadvertently spreading the parasite through your containers, and you should seek guidance from Project Monarch Health or Monarch Joint Venture. At the end of the field season, mail a copy of your Activity 4 datasheet and the sampling card to Project Monarch Health. This information is in your guidebook. Congratulations on completing Activity 4 online training. You can find all training materials and data sheets at the website shown here, or may contact Monarch Joint Venture staff with specific questions at the email address listed below. Thank you, and happy monitoring.